The nucleus is the site at which most of the DNA within a eukaryotic cell resides. A large organelle at around 5 micrometers in diameter, the nucleus is enclosed by an integrated structure composed of two membranes called the nuclear envelope. These two membranes are perforated by thousands of nuclear pores, measuring around 9 nanometers in diameter, providing a physical passage for substances to move between the cell's nucleus and cytoplasm. Each nuclear pore is lined by a massive intricate protein structure, a nuclear pore complex. These complexes are formed by about 30 different proteins called nucleoporins. The function of a nuclear pore complex is to allow selective bidirectional transport, passive diffusion of small molecules and ions, as well as active, regulated transport of RNA, ribosomal subunits, transcription factors, and other macromolecules. This aforementioned active transport is energy dependent. Within the nuclear envelope is the nuclear plasm, faced by the inner nuclear membrane, which contains integral membrane proteins that anchor it to the nuclear lamina and interacts with chromatin and nuclear proteins. Facing out to the cytoplasm is the outer nuclear membrane. This is continuous with the rough endoplasmic reticulum, and like the rough endoplasmic reticulum, it is studied with ribosomes. The outer nuclear membrane is functionally connected to the endomembrane system, a group of intracellular membranes that are either directly connected or communicate via vesicular transport, facilitating protein and lipid exchange. Between these two membranes of the nuclear envelope is the perinuclear space, a gap of around 20 to 40 nanometers wide. The perinuclear space is continuous with the lumen of the endoplasmic reticulum, serving as a buffer zone and potentially playing a role in calcium signaling. The inner nuclear membrane is underlaid by the nuclear lamina, a dense fibrous meshwork composed primarily of lamins, type 5 intermediate filament proteins. It provides mechanical support, assisting in maintaining nuclear shape and helping to anchor chromatin and INM proteins. Inner nuclear membrane proteins with roles including chromatin organization, assistance, nuclear cytoplasmic signaling, assisting mechanical stability of the nucleus, etc. The nuclear envelope is dynamic in nature. Though structurally stable during interphase, the nuclear envelope breaks down during mitosis and reassembles around daughter nuclei during telophase. This is tightly regulated by phosphorylation and dephosphorylation of nuclear envelope components, especially lamins.